c'est ça? Bon, bonjour. I have to introduce myself, he said. So I'm Jean-François Joanny. I am like Uchate. I'm at several places, but they're all within 500 meters. So I have nothing in China. I am at ESPCI. I am a little bit at Curie, and now I have a position at Collège de France, and this is all very close by inside Paris. What I want to do is tell you about tissues, biological tissues. So I will kind of alternate between things that look like active matter and things that look more like biophysics of tissues. And I will try to go from one to the other and see where they meet and what we can do with that. I will give the talk on the board, but unfortunately I'm unable to draw anything on the board. It's immediately infinitely messy. So I have slides to show you figures and movies and pictures and stuff like this. So I will try to alternate. It's not clear to me that it's very easy to alternate. I'm always very happy to come to Les Houches. Yesterday at ESPCI, I met Luca Pelliti, that some of you might know, and he remembered me that we met at Les Houches exactly 40 years ago. So the first time I came to Les Houches was 40 years ago. This was the first year of my PhD, and I was sharing a room with Luca Pelliti, who is still alive and active, and in Paris for a week. So I had to apologize that I would be with you and not with him. So what I want to do in these lectures, I want to discuss the interplay between tissue growth and mechanics. What I mean by that, and I want to try to see how mechanics influences growth, but also how growth influences mechanics. Now, this is not for free. There are at least two domains where this is relevant. One is developmental biology, how organs form, organ morphogenesis. There are stresses during organ morphogenesis, and they influence the final shape of organs. They influence the growth of organs. And the other one, which is more obvious, is cancer. I believe that there is nice physics to do with cancer, and there are lots of people who believe that nowadays. It's kind of obvious that if a tumor grows, it's constrained inside your body, and there are stresses in the tumor. So that was the starting point, and then we started discussing with doctors about that, and most of what I will say comes from this discussion we had at the beginning with these doctors. So I was all the time at Institut Curie at the time, and most of what I know has to do with cancer. I don't know much biology, so I won't use any fancy biological terms. I don't know them myself, so there is no risks that I lose you on that. If by mistake I say a few, please tell me, and the one I will say I can explain. So I call this talk Tissues as Active Materials. I will have to do two things. You really want me to have it?
Yes.
Because I could do without it. Before I start, I will turn the light off. I just want to be a little bit more concrete and show you the type of tissues that we are studying and that I want to talk about. There are essentially three types of tissues that I want to talk about. The first one are these objects. They are aggregates of cells. It's cancer cells. People know how to make them with wild type cells, but they are much more difficult to make. So when physicists do them, they do them with cancer cells. They are called multicellular spheroids. The way they are made is that they start from an aggregate of a few cells, they give it nutrients, and they let it grow. Now, the time scale for these experiments is like 3 weeks, 2 weeks. All kind of weird things can happen. So this looks like a trivial experiment, but nine times out of 10, the whole stuff dies because there's an infection or something like this. So to make a reproducible experiment, it turns out to be pretty hard. These were invented by cancer people, and they are used nowadays to test drugs against cancer, and they are considered as some of the most of the best objects to test drugs against cancer. The difference between all of these is the different methods of imaging which have been used to show them to you. I don't want to discuss that. There are only a few things that I want to show you. So what you see is individual cells. The size of this object is slightly less than 1 millimeter, so it's like 500 microns to 800 microns. This is the size that they reached after 2-3 weeks. And the other thing which is obvious is that they look like sphere. Now, one way to look at that, which is one of the ways I will look at them, is to say they are like liquid droplets made by spheres. So they behave like liquid drops. I will try to convince you that they flow, so they have a viscosity. They are spherical because they have some equivalent of a surface tension. And to me, this is kind of the two main ingredients for the physics of droplets. So that's how I want to look at these things. The second system I want to talk about is the inner structure of the intestine. Now, there is a reason for that, at least two reasons for that. The first one is that the intestine is the organ that your body renews fastest. Within 5 days, all the cells of your intestine have died, and you have divided cells enough that all the cells have been renewed. So in 5 days, because of cell division and cell death, you renew all the cells of your intestine. The other reason is that biologists have a huge number of data on the intestine, and these data are quantitative, so we have things to compare to. Now what you see here is the inner, so the intestine is a tube. Inside the tube it was called the lumen, and what I'm showing you is the inner surface of the lumen, and you see that it's not flat. It has protrusion that goes towards the inside, which are here. These are called villi, and in between these protrusions, here, there are holes that go to the outside, and these are called crypts. The first thing, and this picture is a little bit too small, but if you watch carefully at a picture like this, there is an order in the villi. It's almost a perfect square lattice of villi. Typical size would be a few hundred microns in this direction, and a few hundred microns as the distance between the villi. So that's one of the systems that we studied a lot, because this is, I was going to say, a real biological system. This is the human intestine that we're interested in, and cell division plays a role. It plays an important role, and mechanical instabilities in this case also play an important role. The last type of system that we study is a much more in vitro system. It's what I call confluent cell monolayers. So this is a glass surface on which they put cells. So if you put too few cells, they don't touch each other, they're not confluent. Then they start to touch each other, and they make what people call a confluent layers. So these are cells that fill the whole surface. And then what they do in this case, they invade the surface. So this edge here is progressing in this direction, and it progresses because cells move, and it also progresses because cells divide. You have two types of pictures. This is seen from the side, and this is seen from the top. So this is the edges of the cells that you see here, and here the edges of the cells are here. So these are the three typical types of tissues that I want to discuss. In the beginning, I will tell you things which are a little bit more formal on how I want to discuss them. And then, after 2 or 3 lectures, I will come back to each of these systems and show you what kind of physics can be made with these systems. So the first thing I want, I think I have another one. In the other one, the only thing I wanted to show you is that, because it also gives a nice example of the things I want to discuss. So this is what's called an epithelium. This is a layer of cells that line the surface of an organ. In this case, this is the cervix. So the cervix is here, and there is the epithelium which is here. The epithelium starts here. There is a membrane which is here, and it goes on top of this membrane. The membrane is called the basement membrane, and you see all the kind of things that will be important in the following. The cells which are dark blue here are dividing cells. So you see that cells divide close to this membrane here. They migrate up, so they move up, they go all the way to the top, and they die at the surface here. So this is a type of problems that I want to study. Why do they do that, and how they do that? This involves cell division, it involves cell death, but also in the middle, cells differentiate, which means they change their nature when they go from here to here. In many cases, you start from stem cells, and the stem cells differentiate as they go up. So that's the last example that I want to show you. Roughly speaking, what I want to do is work like a material scientist and say, what about if I consider a tissue like this as a material? Can I do continuum mechanics with that? And what kind of continuum mechanics can I do with that? These are all the examples of continuum mechanics that I know. The tissue can be a solid, and there are obvious examples where it's true. For instance, if you look at plant tissues, they're obviously solids. They have an elastic modulus, and you can characterize the mechanics if you know the elastic modulus. The opposite behavior is a liquid. So for me, a solid is something which has a finite shear modulus, and the liquid is something that flows. And what I want to convince you of is that in many cases, you should look at tissues, in particular at long times, not as solids, but as liquids. And then you have a whole bunch of intermediate behaviors. There's something which is called viscoelastic liquids. These are systems which are solid at short times after you apply a deformation. And as a function of time, they start to flow. And in fact, what I will show you is that tissues are like this. There are also plastic solids. These are solids that flow if you apply a very strong stress on them, like geological fluids, for instance. I won't talk about that, and the reason why I won't talk about that is numerical simulations. I don't have a real explanation for that, but it seems that as long as you have cell division and cell death, the yield stress, which means the stress where they start to flow, essentially goes to zero. So it seems that at least all the tissues that I will talk about don't behave as plastics. They don't behave as plastic because of cell division and cell death. Now, of course, there is something I didn't write here, and which is always at the back of my mind, which is that, and in the sense that Hugo was telling you about, tissues are active systems. So in addition to all of that, the tissue should be active. So that's what I want to show you, how I go from a tissue, which is a very complex system, to a simple description where I describe it as active. So the first thing I want to talk about is the macroscopic or hydrodynamic description of tissues. What I want to do for all of the rest of the lectures is consider the tissue as a living material. And we know, for instance, I don't know if any of you worked in the field of soft matter, that in many cases, if you study complex systems, it's interesting to do hydrodynamic theories. What I mean like that is look at length scales which are larger than the characteristic length scales. So I would look at length scales which are larger than the cell size. And I want to look at large length scale at very long time scales. Now, what fixes a time scale, I told you that I was interested, it goes away every 10 minutes. I want to look at the role of cell division. So at least what I want to do is look at time scales which are larger than the cell division time. Now, the reason why you want to do that when you do hydrodynamic theories is that because in many cases, you don't have to worry about the details. The only thing you have to do is look at the symmetries of the problem. And that's essentially what I will do in the following. Try to build theories which is based on the symmetry of the tissue and not on local details of the cell, which are far too complicated for what I can do. So that's kind of the general idea. Yes, that's what. So, for instance, in the examples I quoted, the cell division time is 1 day. So I won't look at the tissues that I will compress immediately and look to 2 seconds afterwards. I want to look at times which are of order of days. So the tissue is a multi-component system. What I mean by that is that you have cells here, but you have fluids in between the cells. This is called interstitial fluid. This is mostly water, but it's not only water. It's water plus a lot of proteins, plus a lot of polymers. And in most of what I will do in the following, I will replace all that by a tissue as a single component fluid. What this means is that what I will call a cell is the cell itself, plus the interstitial fluid, plus the extracellular matrix, plus everything that goes inside in between the cells. Now you might say that I will lose most of the properties when I do that. It turns out that it's not true. I'll show you on one example that in many cases it's okay to do that. And I'll give you a criterion on how you can do that. As in hydrodynamics, what I will do is define a velocity field. So the velocity field I'll call V of R. It's the velocity of my equivalent single component fluid that, as in hydrodynamics, I average locally. So I do a local average. I define the velocity at each point, and what I will do is study this velocity field using some kind of equivalent hydrodynamic theory. Now, of course, there is a major difference between tissues and classical materials, is that in a tissue, cells divide. So I told you that the time scale for cell division is something like 1 day. It's a complex problem because cell division depends on the environment of the cells. And if you change the environment of the cell, you change the process of cell division. Another thing which is obvious is that a cell won't divide if you don't give it nutrients. So in a tissue, each cell locally consumes energy in the form of nutrients. And for me, this is why it's an active system. An active system is a system that consumes energy locally, and the tissue does that. The energy that you give the tissue is nutrient. Then it's processed inside the cell. Locally, it's consumed in the form of ATP, but in general, what you need to give out to the cells to divide is these nutrients. Of course, the cells need all kinds of things to live. They need nutrients, they need growth factors, they need oxygen, they need all kinds of things like this. This is what I will call the biochemical environment. And what I will do in most cases is imagine that I can make what I will call a chemostat that fixes the biochemical environment, and these variables are all fixed. In certain cases, it's obvious that they play a role, and I give you examples. But in most of what I will do, I will ignore these variables and more focus on the mechanical variables. Now, of course, cells divide, but cells also die. In the my examples have disappeared, but in this system that I was talking about, the main way by which cells die is called bon. Qu'est-ce qui se passe? The main way for cells to die is what's called apoptosis. Now this is programmed cell death. Cells have to die. If they don't die, it's dangerous. And one of the problems of cancer is that apoptosis doesn't occur. So the programmed cell death is not well regulated and cells don't die. So I will assume that they have a rate of cell death and that this is always due to apoptosis. Apoptosis is kind of a clean way of dying. Everything is packaged into small vesicles, the neighboring cells can eat it and things like this. So that's the easiest way by which cells die. So that's kind of my program. I have a non-equilibrium system that requires energy. The system grows because of cell division. It shrinks because of cell death. And what I want to do is look at the mechanics of that. Now I want to go beyond on the board. Yeah, let me try your method. Okay, the first thing I want to talk about is the role of cell division and cell death. Now, of course, where cell division and cell death play a role, it's in the conservation of the number of cells. So that's what I want to discuss now. Ah, merci. So in fact, if I'm more precise, cells grow. When they're big enough, they divide into two. And then the daughter cells grow, and they divide again. Now, if I look at very long time scales, I can put together these two processes, which is cell growth and cell division. And that's what I call cell division. So what I call cell division is the growth plus the actual division of the cells. And the way I will describe that is the most naive way you can imagine. I will introduce a cell division rate. That I call KD. This is the probability per unit time that the cell divides. And I will introduce a cell death rate. That I call Ka for apoptosis, which is the probability per unit time that the cell dies. Now, of course, the conservation law for cells is extremely easy to write. So I call rho the cell density. The cell density is the number of cells per unit volume if it's a 3D system, or per unit area if it's a 2D system. And the conservation law is just dρ dt plus the divergence of V. If the number of cells were conserved, this would be equal to 0. And I have to introduce a source term. This source term is cell division, Kd minus Ka times rho. The growth is due to cell division. Every time a cell divides, there is a new cell. And the loss term is due to cell death. This is what I'm going to call the growth rate. Wait. Yes? Yeah, your question is, do they have a different probability to die? The mother and the daughter, do they have this? In the simple examples, yes. I don't promise you that they're there are no examples where this is not true, which means that the mother will die earlier than the daughter. But in most cases, there is a probability for the cell to divide, and each of the two daughter cells, I mean, there's no mother and there's no daughter, if you want. Both of them are daughters of the initial cells, but in many cases, division is symmetric. There are cases where division is asymmetric. For instance, when a stem cell divides, usually it divides into a stem cell because you need to keep the pool of stem cells, but the other cells could be something completely different. And in this case, cell division is completely asymmetric. And in this case, what you say can be true. The probability to die can be very different for the cells. Yes, so if you want to do it like this, there's a variable, which is a progression in the cell cycle that you can call the age, and I also like to call it the age. What I'm saying is that when the cell divides, I reset the age to 0 for the two cells. And the cell cycle is the same, except in cases where they are very different. The important point is that KD and KA are not fixed variables. So I said that KD depends on the biochemical state. But it also depends on the, yeah. I can't hear. There's a row V missing here. Yes. Merci. That's what you want. That's a current that should be here. So it depends on all the chemistries that I talked about, nutrients, growth factors, certain number of proteins, oxygen, but it also depends on the local density of cells and maybe on the cell pressure. So I will be vague on the cell pressure for a moment, and then this is what I will discuss afterwards. If there is an equation of state, the cell density is related to the cell pressure, or the cell pressure is related to the cell density, and whether I put the cell pressure and the cell density is the same thing. It's not obvious that there is always an equation of state for tissues. Maybe you heard about that. I'm sure you will hear about it at least 10 times, that even for more simple active systems, it's not obvious that you can always define an equation of state. What I will do here is assume that this equation of state exists, so that I have a relationship, whatever it is, pressure as a function of rho. Now, what I will do here is I will kind of ignore these biochemical factors, and what I want to discuss is how these factors, cell division rate and cell death rate, depend on the local pressure or on the local density if it's the same thing. So that's where I want to go. Before I talk about the pressure, I will make a remark. I told you that cell division and cell death are more or less stochastic processes. What this means is that in this equation I can add noise. There is noise because, and I was careful, to talk about it in this way. KD and KA for me is the probability per unit time that the cell dies, or the probability per unit time that the cell divides. So it's a stochastic process, and in this equation there is noise. Now, what I won't show you, but this is well known from population dynamics theory, and you can think about it, and I give that to you as an exercise, that in the limit of long time and large distances, which means in my hydrodynamic limit that I introduced here, the correlation of the noise, so the average value psi of t and r times psi of t prime and r prime, average, is rho, the local density, kd plus ka, delta of r minus r prime times delta of t minus t prime. the assumption that I make when I write that is that cell divisions are not correlated, which means the fact that the cell divides or that the cell die is not related to what happens to the neighboring one. Then if this is true, at long time, the cell division of one cell is only correlated to this cell division, so it occurs at the same position and it occurs at the same time. So if I assume that this is constant, it's okay. But if they depend on rho, that's the next thing I wanted to say. This is multiplicative noise. And then there are all kind of things that you know as well as I do on how should you treat this multiplicative noise. If you discretize, should you take the value at the beginning of the interval or at the middle of the interval? All this plays a role here, and people from population dynamics were very much in advance of physicists on that. They exactly know what to do in this population dynamics problem. You always take the noise if you discretize at the beginning of the interval, because they don't know what happens afterwards. So it's the Ito problem, I think. This is Ito when you take it at the beginning of the interval, yes. I'm always messing these names. So in principle, what I should do is all that. Now if I add noise, I will go from what people call hydrodynamics to generalized hydrodynamics, which is just hydrodynamics with noise. Now this is not the only noise in the system, because if you just walk at movies, cells deform, they fluctuate themselves, the shape of cells fluctuate, and there is a noise associated to that. But at least there is a noise which is associated to cell division and cell death. What I want to do now is discuss the pressure and define what I will call the homeostatic pressure. And to do that, I will go back to my Thank you very much. No, it comes from here. Okay, I want to come to that, which is kind of the first thing we did with tissues, but I still like this picture, and I still think this is the best way to explain what the pressure due to the cells is. So what I want to do, so this is a thought experiment. Physicists love that, so we can do that. This is a box, and the box is permeable to everything the cells needs to live. It's permeable to nutrients, it's permeable to all the proteins that it needs to divide and to die, and it's permeable to water. Now the box, I divide into two, and there's a piston here, and the piston is the same thing. It's permeable to all of that. They are permeable to all of these constituents, but they're not permeable to cells. So I have two compartments. If the cells are in this compartment, they cannot go into the other one. Now, I link the piston to the other side of the cell with a spring. So when the spring is at rest, the piston has a fixed position. And if I push, I compress the spring, and there's a force pushing back. Now, the way I will consider my thought experiments is, suppose I start with a few cells. They are extremely happy, and they divide. So I start from a few cells, they divide, and I have more and more cells in this compartment. At some point, they fill the whole space. So what I told you in the beginning is that they become confluent at this point. But still they want to divide. And if they divide more, they will push the piston. And what I call the pressure due to the cell is the force that they exert on the piston, or if you want, it's the force of the spring divided by the area. Now, I'll tell you first what it is not. There's another way to define the pressure that biologists consider in certain cases, which is to say there's fluid here in between the cells. This fluid is mostly water. There's a hydrostatic pressure in this fluid, and that's the pressure of the tissue. That's not the pressure I want to consider. I'll tell you about that later. But this one essentially doesn't count. It doesn't count because the water can go in and out the cells. So the only pressure that I have is the pressure which is due to the deformation of the cells. So when I compress the cells, they are elastic and they push the piston. You might say this is like osmotic pressure, and that's true. It's very much like osmotic pressure, except that this is a fully non-equilibrium system. For me, the osmotic pressure is a chemical potential. I don't want to talk about chemical potentials in this case, but this is pretty close to that. The other thing that you might have in mind, if some of you know about that, is foams. So if you bubble foams, you have this system with soap films everywhere, and if you compress, water can drain out, but there is a force due to the deformation of the cells of the foam. That's pretty close to that. So that's what I call the pressure. Now, I told you that cell division depends on the local density, and cell death depends on the local density, or on the local pressure. And the pressure has to increase with density, otherwise the system is not stable. So suppose I do this experiment. The cells grow, they exert a pressure on the piston, and they push the piston. If they push the piston, the pressure increases on the cell, and the rate of cell division changes. Now, there are lots of experiments on that, and I'll show you one afterwards. But many of them tell you that cell division decreases if you increase the density or if you increase the pressure. So if you push on the cells, they divide less. So if I do this experiment, they will push here, but they will divide less. But if they push, they increase again the pressure, and so far and so forth, and the cell division rate decreases. Quite similarly, the cell death rate also depends on pressure, and it goes the other way, which means if I increase the pressure, cells will die. So you can say, okay, I can try that. I can put pressure on a single cell. I push. Now, I never saw the experiment, but it's obvious that if you push a cell, you will explode the cell. I think you believe me. If you do that, you need a huge pressure to do that. You need 1 atmosphere or more. If you look at the pressure where cell division and cell death rates start to change, it's much smaller than that. It's like 1,000 Pascal. So the type of pressure that I'm talking about is this pressure of order 1,000 Pascal, over which the cell division decreases and the cell death rate or the cell apoptosis rate increases. Now this goes in the same direction. The cell division decreases, the cell death rate increases, so the growth rate decreases. So if I push the piston more and more, I will reach a place where the cell division is equal to the cell death rate. So maybe I'll write it on the other side. So what I claim, and I will show you experiments, is that cell division he's very, very nice. He will be very tired at the end of the lecture. So cell division decreases with pressure or with rho, and cell death rate, so cell division rate. That's Kd, and Ka increases with pressure. Now if I let this experiment go, and if my box is long enough, the growth rate decreases, and I will reach a state where KD becomes equal to KD becomes equal to KA, so the growth rate is equal to 0. So because the cell division decreases and the cell death rate increases, there will be a pressure where the growth rate goes to 0. Now what it means that the growth rate goes to 0 is that if you lose a cell by cell death, you gain a new one by cell division on average. So it means that the tissue is at steady state. Now this steady state, biologists call it the homeostatic state. It's kind of an equivalent name. So at the homeostatic pressure, cell division equals cell death, and the tissue on average is at a steady state. Now, what I told you is that there is noise. Of course, when I reach this homeostatic pressure, the piston will fluctuate as a function of time. It will fluctuate because by fluctuation at a time t, there will be more cell division than cell death, or more cell death than cell division. So on average, there is a well-defined equilibrium position of the piston, and there are fluctuations around this well-defined position. I have a second exercise for you, which is to calculate the average fluctuation of the piston. So you put the piston here, you put the spring with the spring constant that I call kappa. There is an equilibrium position if the tissue is here, and there are fluctuations. So if I call L the size at the homeostatic pressure, what I want is what is delta L. Delta L on average is 0, so what is delta L squared of T? And for that, you need to use the noise in cell division and cell death that I told you about. Starting from that, there is another quantity that we can discuss, which is, so I will erase that because the next thing I want to discuss is the compressibility of the tissue. So what is the compressibility? It's how much the volume changes when you change pressure. I'm in bad shape today. Okay, so I'll do it another way. I will assume that I have reached this position here. I will move the piston towards another position and block it at the other position. So if I'm at the homeostatic state, whether there is a spring or not, I don't care so much except for the fluctuations, which are not too important. So I can block the piston here, and the tissue will remain at the homeostatic state, and the force that I will have to put on the piston is just the homeostatic pressure. So let me do a naive experiment. I move the piston and I increase the volume. So I increase the volume by delta V. So I fix it here, I move it, and I fix it again. Of course, if I move it, I create a gap where there is no cells, so cells will divide. cells will divide and will fill the gap. The pressure will increase again on the piston. In the beginning it's 0, it will increase again. The pressure will increase, and it will increase until I reach a steady state. So it will increase until I reach again the homeostatic pressure. So what I'm kind of telling you indirectly is that I change the volume and I get the same pressure. Everybody agrees with me? So I'll tell you again how I do the experiment. I reach the homeostatic state, I block the piston, everything stays at the homeostatic state, then I move the piston further away. There's a gap, the cells will fill this gap and try to reach a new equilibrium. This new equilibrium will be at the homeostatic pressure, so the same pressure that I started from. I get rid of the spring. I just say I block the piston, I push it, and I block the piston again. And it stops at the homeostatic state, otherwise I wouldn't reach an equilibrium. So it means I can change the volume without changing the pressure. So it means a tissue like this is infinitely compressible. So for a theoretical physicist, it's nice. I found a system which is infinitely compressible. Do you know any other one? A system, a liquid-gas equilibrium at the critical point. There are fluctuations between gas that can transform into liquid and liquid that can transform into gas, and that's also infinitely compressible. However, I need to be exactly at the critical point to be infinitely compressible. So I need to fine-tune the system perfectly so that the compressibility is infinite. Here I don't need to do that. Whatever the initial volume, I will reach the same thing. The only steady state that I have will be the homeostatic state. The other question, which is more relevant, is whether I know of any biological question where this plays a role, and the answer is no. So what I'm claiming is this is a nice idea, but I don't think it's of any use. And in practical situations, there's always something that makes the system compressible, and there's no big effect. Another thing you can guess, because you did a lot of thermodynamics, is that if the tissue is infinitely compressible, and this is true at the critical point, there are huge fluctuations. And that's true. Now in my exercise on the piston, if you go there, the position of the piston diffuses. So there's such sufficient fluctuation that the position of the piston fluctuates, and the amplitude, the square of the amplitude increases linearly with time. So that's associated to this infinite compressibility, but this is the only problem that I know where it could play a role. So it's kind of a gadget for physicists. The next thing I want to do is try to show you how to measure the homeostatic pressure. Now, when we started doing that, we were pretty ambitious. And the idea that we had is that when people talk about cancer, for instance, they have all kind of pathways that they know that will induce cancer. And then if you want to know what happens in a tumor, you have to measure these pathways. One of our ideas was, okay, so why not measure the homeostatic pressure? If it's big, maybe the tissue invades more of the neighboring tissues, and that would give you an idea of the aggressiveness of the tissue. So immediately we went to our friend experimentalists and we told them, okay, why don't you measure the homeostatic pressure? So there are experiments in the literature where this is done. And the most clever one is this one. These are people that work with gels. So these are soft gels which are water soluble. They are filled with water. They are soft elastic material. They make a hole in the gel and they put cells in there. The cells grow. The tissue, the aggregate of cell grows. At some point it reaches the edge of the hole and it pushes on the gel, and it creates stresses in the gel, which is like the pressure I was applying. And at some point, it stops to go. Now, this is what these people did. This is a group of Jane in the US. And in fact, they had a surprise. The surprise that they had is that the homeostatic pressure is strong enough that it cracks the gel. So the gel doesn't only compress, but it cracks locally. And it cracks here. So this part is compressed by the gel, and this one is not compressed by the gel. And then what they did is they measured cell division and cell death. The only pictures I have are on cell division. So the green cells are the ones which divide. The pressure is lower here, and the cell divide where the pressure is lower. The pressure is stronger here. The cells divide less. And if you do the symmetric experiments with cell death, cells tend to die where the pressure is higher. So this is one of the experiments that tells you that cell division decreases with pressure and cell death increases with pressure. So this next experiment is one we did in Curie. I'll tell you about it when I talk about multicellular spheroids. There is an experiment, I don't even know why I put it here, but that I find very nice, which is an experiment of Françoise Brochard. When we told her the story about spheroids, she said, but it's like liquid drops, so I should be able to do wetting experiments with that. And the experiment that she did is this one. She takes a micropipette, she swallows the edge of the spheroid here, and she lowers the pressure here. So the spheroid goes a little bit in there. She waits, then she decreases the pressure, and the spheroid goes little by little in the micropipette. So she decreases the pressure, and you will see a tongue forming here, which again tends to tell you that these tissues behave like liquids, which is kind of what I'm after. Then we had very long discussions on how could we measure this homeostatic pressure in vivo, because if you want to do it, you want to do it in a real tumor. I remember having a discussion with a doctor who told me, okay, I kind of understand what you say, but I know how you test it. I know all the cancers, all the human cancers you can induce in mice, and I know which one are aggressive and which one are not aggressive. So you figure out a way, I'll give you mice with all these tumors, and you figure out a way to measure the homeostatic pressure, and then I'll tell you whether it's true or not that it measures invasiveness. There are two ideas. We are not there yet. It's several years and we are not there. The first one is due to Giovanni Capello, who was in Curie and now has moved to Grenoble. And his idea is very simple. He says, let me take small pieces of very soft gels and I put it inside the tissue. If I change the pressure locally, I change the stress on this particle, and if I increase the pressure, it will get compressed. If I decrease the pressure, it will dilate. So that's what he did. He put these green pieces of gel, changes the pressure, and look how these beads change volume. The idea is nice. The problem is you have to, if you want to do it in vivo, you have to be able to measure in vivo the volume of the gel, which is non-trivial. And the other thing is you are not sensitive to very low pressure differences. So if you apply a huge pressure, you change the volume. And if you want to be more sensitive, you need gel with a much weaker elastic modulus, and that's more and more difficult. So in principle, this is a nice experiment. He's able to do it on a spheroid, but he's not able to do it in vivo. There's another idea, which is due to O.J. Campas, who is another person who was in Curie, who at the time was a theorist, but he became an experimentalist. And his idea was that instead of putting gel, so elastic material in the tumor, you can put oil droplets. Now, oil droplets are elastic because if you deform them, you increase the area and you increase the surface tension energy. So there is an elasticity of these oil droplets, which is directly proportional to the surface tension. It's even the surface tension times the number. So he did that, but again, it's not sensitive enough. What he's able to do is to measure deformations under shear. So under shear, it's easy to do. There's no way you can do it under compression. So he even tried with air bubbles, but he wasn't able to do the measurements. So the idea is good, and you can do it on all kinds of tissues. You have to put an object that gets deformed, and you have to be able to measure the deformation of this object. Qualitatively, it works. Quantitatively, it doesn't work with this gel, and it doesn't work with these oil droplets. So that's where we are. And there's still a lot of work to do, and maybe to be very clever to find one of these, to find a method to. Chemical, or something like light that would change, the wavelengths would change when you compress, or something like this. Yes, I don't know. They never tried that. Maybe it's a good idea. You need something which is extremely sensitive to very low pressure difference. That's what you need to find, and something that you can measure. Even this stuff in the mice is not very easy, because you can do it only very close to the surface. Maybe with ultrasounds you could do it better, but that's not so easy. Yeah, I have a last general thing that I want to say, and then I'll go to more quantitative things. What happens is now if you take two tissues and you put the two tissues in the same space. So I do the same experiment. This is my piston that can move inside. But now I suppress the spring and I put two types of cells, one on each side. So I have a cell, a tissue that I call a cancerous tissue. I will show you in a second, you will understand why. And on this side, I put what I call the healthy tissue. I put the compartment in the middle, and I start with a few cells on each side. Now again, the cells will divide because they are free and happy. They will divide, and they will become confluent on each side. Now, of course, these two tissues have no reason to have the same homeostatic pressure. So at some point, I will raise the homeostatic pressure of the lower tissue. The piston is at equilibrium, so the pressure is the same on the other side, but the pressure on the homeostatic pressure is higher on this side. So on this side, the cells are happy, they want to remain in a steady state. On this side, the pressure is low, so the cells want to divide. But if they divide, they push the piston in this direction, they increase the pressure on this side, and the cells die on this side. So another way to say that is that if I have two different tissues in a confined volume, there's always one tissue which wins. It's the tissue where the homeostatic pressure is larger. There is no stable steady state where the piston is in the middle. Either it goes to the right because the pressure is larger on this side, or it goes to the left because the pressure is larger on this side. Do you all agree with that? And that's independent of the model that I will use for the dynamics. It's just a question of pressure. It doesn't depend on the fact that in the beginning cells divide faster on this side or faster on this side. The only thing that matters, and that's independent of the model, is which of the tissues has a higher homeostatic pressure. And this one invades the other one. It invades the other one in the sense that the other one disappears because all the cells die. Sorry? I'll show you an answer to that even worse than that. But wait, I don't know, tomorrow or the day after tomorrow. Now, what I will show you is numerical simulations. We have a postdoc, Jens Elgeti, who set up a simulation program. I'll show you how it works afterwards. What he does that, I don't know if you see, there's a spheroid, an aggregate of cells here, which occupies a given volume. So it's blue, and it's dark blue here, and you start from one red cell in the middle. But the pressure, the homeostatic pressure of the red cells is higher. And what you see is that the red cells invade everything, and this is seen from inside. This is the blue cells that die. So if two tissues compete for space, in this simulation, as well as in this argument here, the tissue with the highest homeostatic pressure invades the tissue with the lowest homeostatic pressure. Whatever model you use, you have to end up with that. This is why we're thinking that this could be useful for cancer. As we are not so much able to measure it, it's not very useful yet, but who knows. If there is no, if the volume is not constrained, this doesn't happen, and the tissue phase separates. There's one that goes inside and one that goes outside. Maybe I'll discuss that afterwards. So, okay, so these are a few general things. What I showed you is that cell division decreases with pressure and cell death increases with pressure. There is this pressure that I call the homeostatic pressure, where the tissue is at a steady state. And if I have two tissues, the one that invades the other one is the one with the highest pressure. What I want to do now Merci. J'ai tout perdu là. C'est mes oreilles à moi que j'ai perdu maintenant. OK. So what I want to talk about is the fluidization of tissues by cell division. Now what I want to discuss is a very simple model for the mechanics of a tissue. If I take a cell and I push on a cell, I told you that there's a well-defined shear modulus or Young modulus. It's pretty low. It's 1000 Pascal. Who can tell me something which has a modulus of 1000 Pascal? How much is this? Hundreds of gigapascal. It's much softer than that. How much is this? My shoes or some elastomers? Megapascal, yeah. Maybe a bit more, maybe a bit less, but the order of magnitude is okay. So what else do I know? Sorry? Hydrogels. Yes. So the one I like to talk about, I don't know how many of you are English, except for Julia, but she knows my story. English like to make these jellies in the morning. The stuff, they put it in your plate and it not in the morning. This has a shear modulus of 1000 Pascal. So it's extremely soft. And that's the softness of or the elastic modulus of a cell. Now, if you take a tissue and you apply a shear on the tissue, at very short times, it has an elastic modulus, and the elastic modulus is more or less the elastic modulus of the cells. If I want to look in more details, it's a little bit different because the patting matters and things like this, but it's the same order of magnitude. So if I apply a shear, a stress, at short time I have an elastic response. And if I create a deformation, I have an elastic stress, so that I will call sigma alpha beta elastic. The sigma alpha beta elastic is the shear modulus times the factor of 2 times the deformation. What I'm claiming is that this is not the only stress in the tissue, and these are not the only forces in the tissue. When a cell divides, what I told you in the beginning, that what I call cell division is cell growth plus cell division. So what it means is that when a cell divides, it has to push on the neighbors. When a cell dies, the volume shrinks, and the neighbors can go in, so it pulls on the neighbors. And that's what I want to take into account. So we'll see. But instantaneously after I push, you agree that there will be a deformation which is kind of the same as the cell. I agree it's a little bit different, but it's hard to calculate the difference because it depends on the packing of these soft objects. But maybe it's 2000 Pascal or 500 Pascals, I don't care so much. But there is a well-defined shear modulus of the tissue. So a dividing cell pushes on the neighbors. Now, how should I describe that? The claim that I want to make is that a dividing cell cannot exert a force on its environment, because if it exerts a force on its environment, the environment exerts the opposite force, and the cell is not at mechanical balance. So if you want to describe the effect of a cell on its environment, at least it has to be a force dipole, because the total force has to be 0. So I will do the simplest thing that I can do, is assume that when a cell divides, it exerts a force dipole on its environment. Now, a force dipole is you have 2 points like this, and you have 2 forces, F minus F, and there is a distance D between the 2 forces. So that's what the force exerts on its neighborhood. And then there could be higher order multipolar terms. Yes? Cell division. Yeah, okay. No. Okay, I'll go back to the first thing I said, is that the way I describe it, cell division and cell growth are the same process. Okay? I agree that what pushes on the neighborhood is cell growth. Maybe cell division a bit, but mostly cell growth. And cell growth is important when, before dividing, the cell reaches twice the volume that it has when it doesn't divide. So that's an important effect. Okay? So how do I measure a force dipole? I measure it with a tensor that I call d alpha beta, which is f beta d alpha, where alpha and beta are the Cartesian components of the force, which is here, and on the distance between the two forces where the 2 points where the force is applied. So that's the local force dipole. Now, of course, the environment reacts. The environment reacts because it has an external force applied on it, which is a force dipole. So if I want to write the total force balance, so the divergence of the stress plus the sum of the forces will be equal to 0. So d alpha sigma elastic alpha beta, that will be the elastic force. And then for each dividing cells plus sum over I, which are the dividing cells, of F, so I will call this point R. So F of Fi of R plus di minus so the force of the cell I has a force applied at point R plus dr here, or I could it and minus a force applied at point R. Now this, if I want to write it more correctly, is plus sum of Fi delta of R minus Ri plus di minus Fi delta so I have to sum all the forces. This is kind of written in a weird way, but I have to sum all the forces exerted by the dividing cells. Each dividing cell exerts two forces, a force plus Fi, which is at position Ri plus di, where Ri is the position of the cell, and the force minus Fi, which is at position Ri. Okay? Now, I will rewrite that. I will assume that di is small and just expand that in powers of di. So this is minus Fi beta d beta d alpha, sorry, of delta of R minus Ri times di alpha. Is that okay? Yes? No? I can redo it. I kind of wrote this, which is not very correct. What I just wanted to say here is that I have to look in the fluid which is the force exerted by cell I, and there are two forces, one at position Ri plus di and one at position Ri. Okay? And here I just expand this expression here. So this is just, if I write the force balance here, d alpha plus minus d alpha f beta d beta of R minus Ri. And that should be equal to 0. The total force, local force, should be equal to 0. Part of the force comes from the stress, and part of the force comes from the forces due to each dividing cells. Ah, yes. So what he's saying is this term symmetric? So I have here, I have d alpha f beta, and you would want me to write d alpha f beta plus d beta f alpha. But the force dipole is symmetric, otherwise there would be a torque. So if there is no torque, if the cells are not chiral, there is no torque, and I know that it's symmetric. Okay? So this is why I drew it like this. If I draw it like this, there is no torque. If I would draw it like this, there would be a torque, and then the torque wouldn't be balanced. So yes, it's symmetric. It doesn't appear here, but it's exactly symmetric. Okay, so now what I can do is I can introduce the force dipole density. And I have a sum over I. that I will call D alpha beta. Alpha is here. Je suis un peu endormi. And that's it, no? No, the beta is here, the delta is missing. Yes. Okay, I agree. D alpha is here. Voilà. I agree. So here as well, no, I have D alpha here. Okay. So I introduce the force dipole density, which is sum of di alpha beta delta of r minus ri. So I count the force dipole at each point where there is a dividing cell. And what I have here is just a derivative of this force dipole density. So the force balance is d alpha minus d alpha times the force dipole density is equal to 0. So what this tells me is that inside the tissue, I have the elastic stress, but I have another stress, which is an internal stress due to cell division, and this internal stress is minus the force dipole density. All the ones which are currently dividing. And I have the same contribution for all the cells which are currently dying, which I want to do. Dividing meaning growth and dying meaning shrink. It's a little bit less obvious for the dying cells, but for dividing cells, they push on their neighbors. Yeah, so then what I would have to do is go into how does the volume change when a cell divides. That's not so well established. I mean, I could accept that it's kind of constant and then it starts to grow when it duplicates the biological material. And that's what I would call a dividing cells. You are kind of saying, anyways, they're all in the process of dividing. I'm doing it some kind of average here because I assume that all the cells will have the same dipole, force dipole. I don't think, if I were looking exactly on cell divide, what you say would be important. But if I look at long times, I don't care so much what I call the dividing cells. The only thing which is important is by the process of cell division and cell death, I'm created a stress inside the tissue. And if I want to write the force balance inside the tissue, I have to balance this stress with the elastic stress. Did everybody get what Ricard was saying? He's saying, I told you that I sum over all the cells which are dividing. But he says a cell is always growing. In almost all the steps of this cell cycle, it's growing, but it's growing more or less. So I have to define some kind of criterion of what I call a dividing cell, and then I average on that. Okay? So I define what I will call the internal stress. Sigma internal alpha beta, which is equal to minus the force dipole density, and the total stress in the tissue is sigma alpha beta equals sigma elastic alpha beta plus sigma internal alpha beta. Okay, so what I want to do now is discuss how this total stress is related to the deformation in the tissue. And the relation between the total stress and the deformation is called the constitutive equation of the tissue. Now to do that, I will split the stress into two components. One which is the trace, the diagonal part of the stress, and one which is the part of the stress which has zero trace. So what I want to do is write that the total stress sigma alpha beta, I will write it as something which is diagonal, sigma delta alpha beta, plus sigma tilde alpha beta, and I will choose sigma in such a way that this sigma tilde has zero trace. So I will impose sigma alpha alpha equals 0. Now this sigma is simple. It's minus the pressure. The pressure is related to the opposite of the diagonal part of the stress. So sigma equals minus P. And this one doesn't include the pressure. It's traceless. A shear, for instance, would have only this component. An isotropic deformation would have only this component. And what I want to do is discuss independently this term and this term. So I will discuss the isotropic stress. It's not obvious. Now, as I told you in the beginning, what I'm kind of after is whether a tissue is rather liquid or solid or viscoelastic. All of this for me is a constitutive equation, which means it depends on u alpha beta, but also on its derivative. And in this case, I found one. Now, from a priori, it's not obvious to me that there should be a direct relation. You can also worry at long times how is u alpha beta defined, because cells divide, cells die. So if you watch at long time, you don't have the same cells that you had in the beginning. So yes, that's possible. I will show you that with only the hypothesis that I made, I will reach something simple. But all this is possible. It's like a granular material. Is it even obvious that you can define u alpha beta? It's not entirely obvious, but for practical purposes, it seems like a good idea to define it. U dot of u will count. I mean, when I say u alpha beta, it could be u dot. That, for me, is the same thing. It's u as a function of t, and it's not obvious that it's local in time, for instance. Okay? So I start with this sigma, or with the pressure, and I say this sigma has two contributions. Sigma is sigma internal plus sigma elastic. So sigma elastic is the elastic deformation just after I apply the stress. And for that, I will lose classical linear elasticity. So sigma elastic, so I have to look at the sign, is K times U alpha alpha. It's just due to the compression of the cells by an anisotropic pressure, where U alpha alpha is a divergence of is a trace of the deformation tensor. Sorry? At long time scale is 0. I agree. But at short time scale, I have this, and I should have something like this. Okay? Now, if I take the derivative of that, and I will take the convected derivative of that, I will write that d sigma elastic dt is k d dt of u alpha alpha. Now the derivative of the displacement is the velocity. So I will introduce the velocity gradient. Or if you want, what's called the shear rate tensor in hydrodynamic. d dt v alpha beta equals 1/2 of d alpha v beta plus d beta v alpha. The trace of that v alpha alpha is just the time derivative of u alpha alpha. So this is k v alpha alpha. Okay, that's the easy part. This is the bare elasticity of the cells. What I want to do now is discuss the internal stress. Now maybe related to the question of Ricard, the absolute value of the internal stress is not interesting. What is easy to track is how the internal stress changes with time. Because what I define for cell division is a rate of cell division. So the only thing that I will be able to define properly is how does the internal stress changes with time. So I want to write d sigma internal dt. Now this is due to cell division. So I will write that it's proportional to the number of cells, and it's proportional to the rate of cell division. Now, when the cell divides, it exerts a pressure. The pressure is positive, so the stress is negative. So I will put a negative sign here. Now, the pressure that the cell exerts is related to this force dipole that the cell exerts on its neighborhood. So I will put a phenomenological value of this pressure that I will call d division. So I say the internal stress changes because cells divide. The probability that there is a cell dividing is rho times kd, and then the change in stress to the one division I call d sub d. Now I have a term like this, and then I have the equivalent term due to cell death, plus Ka dA. And you will agree with me that if it's a pressure when it divides, it's a negative pressure when it dies. So this dA has the opposite sign of dd. Okay, now see, if I want to plug everything in here, I can calculate d sigma dt. So it's k u alpha alpha minus rho I have a positive, the cell has to push on the neighbor. So I create a positive pressure and the stress is minus the pressure. Okay? So the cell that divides is equivalent to a positive pressure on the rest. So it's a negative stress. And there is the opposite sign for the cells that die. So this one should be the opposite sign. Now, suppose that I assume that the tissue has an equation of state. Sorry? It's V now. That's the divergence of the velocity. Suppose I assume that there is an equation of state. If there is an equation of state, I have a relation between sigma and the local density. So my equation of state is something like sigma as a function of rho. Now from sigma as a function of rho, I can calculate d sigma dt. So d sigma dt is d sigma d rho dt. Sigma depends only on rho, and if rho changes with time, sigma will change with time according to that. But d rho dt I know because of the conservation law of the cells. So I write that back. D rho dt plus divergence of rho v is kd minus ka. So if I want to write the convected time derivative d rho dt, I take the term v dot grad rho and I put the other one on the other side. So this is kd minus ka that comes from here, and there is a term that comes from here which is rho minus rho divergence of v. But d sigma d rho is just the compressibility. So I have to write it the right way. D sigma d rho is minus k over rho, where rho is the compressibility. So here again, there is a minus sign because sigma is minus the pressure. On arrête. Let me just plug that. Okay, ask your question. Rho is missing here. Okay, so I can write d sigma dt from here is k kd minus ka minus k over rho divergence of v. Is that what I wanted to write? Yeah. Okay, did I put the right sign where I needed to? No, it's a plus here and a minus here. Okay. Z minus. Okay, so I'll stop here and I'll show you what it gives tomorrow afternoon.